Hello students, in this lecture video, let us understand the structure of proteins. We already know what proteins are. These are heteropolymers of amino acids. Now to begin with, firstly let us look at the primary structure. It refers to the amino acid sequence which is at the N-terminal as well as C-terminal end of a protein. So this chain it looks like, starting with glutamic acid, alanine, valine, glycine, leucine, valine again, serine and phenylalanine. Then coming to secondary structure. In secondary structure, the proteins, these amino acids in the proteins are folded. So right-handed helices are generally found in folded proteins. Next is tertiary structure, the next level of folding, in which this long chain folds upon itself to give a three-dimensional structure. The last type of structure which is formed by folding is quaternary structure. In this, the proteins are assembled with subunits which have more than one polypeptide, like in hemoglobin. So these structures contain certain bonds which are called as peptide bonds, which is formed by hydrolysis. Basically, there is loss of water when one carboxyl group of one amino acid reacts with amino group of the next amino acid in the peptide chain. So it forms CONH bond, which is called as peptide bond. Next is glycosidic bond, which is formed when one sugar reacts with other sugar, forming a glycosidic bond. Next is phosphodiester bond, in which there are two ester bonds with the phosphate group. That is, we are talking about nucleic acids, in which the nitrogenous base is linked with the pentose sugar, and the O molecule in this sugar reacts with phosphate group. So two ester groups are formed with phosphate. That's why phosphodiester bond. Next, nucleic acids, they exhibit variety of secondary structures. How? Because again, it is a double helical structure like in proteins, and these nucleotides are arranged or stacked one on the other, giving it more stability. When it forms a helix, the one turn which is formed in this Watson-Crick model shown over here, the distance is 3.4 angstrom, which is also called as pitch of helix. The Watson and Crick model actually explained the B type of DNA, which is right-handed and in which the pitch is 3.4 angstrom, which equals 10 raised to the power minus 10 meters. Next, coming to the concept of dynamic state of body constituents. See children, all the biochemicals which are there, biomolecules in our body, these are always moving. These are not stationary. So their concentration varies and their turnover varies. What it is? Means the concentration can be measured in moles per cell or moles per liter. Turnover means that one biomolecule is constantly being changed into another. It is not stationary. And metabolism, we all know by now, is the total chemical reactions which are taking place in the body, which means that the biomolecules are transforming into each other. Means there is transformation. Nothing is stationary. It is a flux. So these metabolites, these are converted into each other with a series of reactions called as a metabolic pathway, which can be linear, it can be circular or crisscross. Means it can be converted into a variety of metabolites which are linked in different pathways. So this flow is in dynamic state. And every reaction in the body is catalyzed in living system with the help of enzymes. Now coming to two types of pathways, anabolic and catabolic. Anabolic, in which the simple molecules convert into complex, and catabolic, where complex degrades into simple molecules. In anabolic, because complex molecules have to be formed, so it will consume energy. Whereas in catabolic pathways, as simple molecules are formed, they will release energy, which is generally in the form of ATP, that is adenosine triphosphate. Very common example, glycolysis. Now these concentration of biomolecules vary. Like in blood, the concentration of glucose is in millimolars, whereas of hormones is in nanograms. So that is the variety of concentration. So, by this, we understand that biomolecules are always in a flux, means they are not in any equilibrium state, it is in steady state. Steady state helps to perform work with constant effort, energy input, which is directly proportional to the living state.